Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Gene therapy tools: types, methods, applications, advantages, and disadvantages. Table of contents. Introduction to gene delivery method. Types of gene delivery methods. 1. Nonviral transfection methods. A. Liposomes mediated. B. Biolistics Gene Gun. 2. Virus-mediated transduction methods. A. Retrovirus-based vector. B. Adenovirus-based vector. C. Adeno-associated viruses-based vector, AAV. Advantages and disadvantages of gene delivery methods. Introduction to gene delivery method. One of the primary challenges in somatic gene therapy is identifying an effective gene delivery system. Ensuring that the target tissues receive the appropriate genes is crucial for the success of the therapy. Gene therapy can involve the direct introduction of naked DNA into target cells. However, this method has a very low efficiency rate, as isolated DNA molecules are not easily taken up by the cells. To improve the efficiency of DNA uptake by target cells, specially engineered vectors are used for gene transfer. These vectors are designed to facilitate the delivery and integration of therapeutic genes into the target tissues. Developing appropriate methods to deliver DNA is vital for the success of gene therapy. Effective delivery systems ensure that the therapeutic genes reach the target cells in sufficient quantities to produce the desired therapeutic effect. By leveraging advanced vectors and delivery methods, the efficiency and success rate of gene therapy can be significantly enhanced, paving the way for more effective treatments. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Types of gene delivery methods. There are two primary gene delivery methods. 1. Non-viral transfection method. 2. Viral-mediated transduction method. 1. Non-viral transfection methods. Currently, two non-viral transfection methods, liposome and biolistic, have been tested for in vivo or ex vivo gene therapy approaches. A. Liposomes mediated. In liposome-mediated gene delivery, the DNA to be transferred is packaged in vitro with liposomes and then used directly for gene transfer to target tissues in vivo. Liposomes can be either cationic or anionic. Anionic liposomes have a negative surface charge that helps in binding DNA inside. The lipid coating of liposomes protects the DNA in vivo, facilitates its binding to target cells, and aids in the endocytosis process, allowing the DNA to enter the cells. Advantages: no viral components are involved, reducing the risk of pathogenicity. Non-pathogenic, minimizing health risks associated with the delivery method. No immunity problems, as liposomes do not typically provoke immune responses. No limit to the size of the foreign gene that can be delivered, offering flexibility in therapeutic applications. Enhanced stability and protection of DNA from degradation in the extracellular environment. Potential for repeated administrations without the risk of immune sensitization. B. Biolistics Gene Gun. In the biolistic method, high-velocity DNA-coated microprojectiles are accelerated into the target cells. The process involves using a gene gun to shoot tiny particles, typically gold or tungsten, coated with DNA into the cells, enabling the DNA to penetrate the cell membrane and enter the cellular environment. Advantages: no viral components are involved, reducing the risk of pathogenicity. Non-pathogenic, minimizing health risks associated with the delivery method. No immunity problems, as the method does not typically provoke immune responses. No limit to the size of the foreign gene that can be delivered, offering flexibility in therapeutic applications. Direct delivery into the cells, potentially increasing the efficiency of gene transfer. Disadvantages: potential for physical damage to the target cells due to the high-velocity particles. Requires specialized equipment, such as a gene gun, which may not be readily available in all settings. Limited to accessible tissues and may not be suitable for all types of tissues or organs. 2. Virus-mediated transduction methods. Viruses introduce their genetic material into host cells to hijack the cellular machinery and produce new viruses. This genetic material contains instructions for the host cell to generate viral components. Besides the genes for virus production, viruses can also carry additional genes for synthesizing other proteins. Three main types of viruses are commonly used in gene therapy: retroviruses, adenoviruses, and adeno-associated viruses. These viruses differ in their mechanisms of action and outcomes. A retrovirus-based vector. Retroviruses contain their genetic material in the form of RNA molecules, whereas the host cells have their genetic material in the form of DNA. When a retrovirus infects a host cell, it introduces its RNA and enzymes into the cell. The retroviral RNA must be converted into DNA molecules, a process called reverse transcription, carried out by the enzyme reverse transcriptase. Once the DNA copy is produced, it integrates into the host cell's genome, facilitated by the enzyme integrase. This integration means the viral genetic material becomes part of the host cell's genetic material. As the host cell divides, its descendants inherit the new genes introduced by the virus. A significant challenge with retrovirus-based gene therapy is that the integrase enzyme can insert the viral genetic material at random positions within the host genome. If the viral DNA is inserted into the middle of a functional host gene, it can disrupt that gene. This disruption can have serious consequences, such as interfering with genes that regulate cell division, potentially leading to uncontrolled cell division and cancer. Strategy for retrovirus-based vector-mediated gene delivery. The most commonly used retrovirus-based vector is derived from the murine leukemia virus, MLV. MLV can infect both mouse and human cells, but it only targets rapidly dividing cells. Vector development involves deleting the essential genes GAG, POL, and NV from the retrovirus, making it replication deficient. Deleting these genes creates space for inserting an expression cassette containing the therapeutic gene of interest. The modified retrovirus, now containing the therapeutic gene, serves as a vector for delivering the expression cassette. Despite being replication deficient, the modified retrovirus remains infectious and can effectively deliver the therapeutic gene to target cells. Advantages of retrovirus-based vector: integrate into the host cell's genome, providing stable and long-term gene expression. Contain no viral genes, reducing the risk of viral gene-related complications. Cause few immunity problems, minimizing immune responses against the vector. Disadvantages of retrovirus-based vector: can only infect dividing cells, limiting their use to tissues with actively dividing cells. Random integration may cause insertional mutations, potentially disrupting important genes and leading to adverse effects such as cancer. B. Adenovirus-based vector. These DNA viruses have a linear double-stranded genome of approximately 36 kilobase pairs, kbp. When adenoviruses infect a host cell, they introduce their linear double-stranded DNA molecules into the host genome. However, the genetic material of the adenovirus does not integrate into the host cell's genome. Instead, the linear double-stranded DNA genome of the adenovirus remains as an episome within the cell nucleus. The genes carried by these extra DNA molecules are transcribed similarly to host genes. These additional genes do not participate in cell division, meaning that as the host cell divides, its descendants do not inherit the extra genes. Therefore, therapeutic applications using adenoviruses often require repeated administration to ensure that the missing gene is continually supplied, as the episomal DNA does not persist in the host cell's genome. Advantages of adenovirus-based vector: can accommodate large DNA inserts, up to more than 30 kilobase pairs, kbp, allowing for the delivery of complex genetic constructs. Infect both dividing and non-dividing cells efficiently, broadening their applicability in gene therapy. Disadvantages of adenovirus-based vector: do not provide long-term gene expression because their DNA does not integrate into the host cell genome, leading to episomal maintenance that is not stable over time. Highly immunogenic, triggering strong immune responses in the host, which can limit the effectiveness of the vector upon repeated administrations. See Adeno-associated viruses-based vector, AAV. Adeno-associated viruses, AAV, belong to the parvovirus family, comprising non-pathogenic single-stranded DNA viruses. They require co-infection with adenovirus or herpes virus for replication. Despite their advantages, AAV vectors have limitations such as their small carrying capacity of approximately 4.5 kb and challenges in production. However, AAV is non-pathogenic, with many individuals carrying it harmlessly as a latent virus. Unlike adenoviruses, AAV vectors often provoke immune responses in treated individuals, which can lead to clearance of the virus and the cells that have been successfully treated with it. Advantages of gene delivery methods. Integrate into the host cell's genome, specifically chromosome 19, and provide stable expression of the delivered genes. Do not contain any viral genes, reducing the risk of complications related to viral gene expression. Non-pathogenic, posing minimal health risks when used in gene therapy applications. Cause no significant immune response issues, making them suitable for repeated administrations. Infect both dividing and non-dividing cells effectively, broadening their utility in treating various cell types and tissues. Disadvantages of gene delivery methods. Have a limited carrying capacity for foreign genes due to their relatively small genome sizes, approximately 4.7 kb, restricting the size and complexity of genes that can be delivered. Present challenges in obtaining large quantities of viral stocks, which can hinder their widespread use and scalability in gene therapy treatments. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Advantages and disadvantages of gene delivery methods. Advantages of gene delivery methods. Stable integration and expression. Some methods, like retroviruses and AAV, integrate into the host genome, providing stable and long-term expression of therapeutic genes. Nonviral. Many gene delivery methods, such as AAV and non-viral vectors like liposomes and biolistics, do not involve viral components, reducing the risk of pathogenicity and immune responses. Wide applicability. Gene delivery methods can infect both dividing and non-dividing cells, expanding their applicability to various tissues and cell types. Controlled immunogenicity. Non-pathogenic vectors like AAV are generally well tolerated and induce minimal immune responses, allowing for repeated administrations if necessary. Disadvantages of gene delivery methods. Limited cargo capacity. Viral vectors, especially AAV, have a limited carrying capacity for foreign genes, which can restrict the size and complexity of therapeutic genes that can be delivered. Integration risks. Vectors that integrate into the host genome, such as retroviruses, pose a risk of insertional mutagenesis, where the integration may disrupt normal cellular genes and potentially lead to adverse effects such as cancer. Production challenges. Some viral vectors, like AAV, can be challenging to produce in large quantities, which may limit their widespread use and scalability in gene therapy. Immunogenicity. Concerns. Despite efforts to minimize immune responses, some vectors can still provoke immune reactions in patients, potentially reducing their effectiveness over time or upon repeated administrations. Navigating these advantages and disadvantages requires careful consideration of the specific therapeutic goals, target tissue, and patient characteristics to optimize the effectiveness and safety of gene delivery methods in clinical applications. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!